We have recently discussed meiosis, and meiosis is the division of our cells to produce our haploid reproductive gametes. Remember, gametes are sperm and egg cells that have a reduced number of chromosomes. Because of the events of meiosis, it contributes to genetic variation. But there's a few events that we want to highlight in reference to genetic variation. We've already seen crossing over, but remember that crossing over is the exchange of information on homologous chromosomes. Remember, they're just trading places. It's a fair trade. And since these genes do belong on these chromosomes, it is not problematic. This occurs during prophase 1 of meiosis 1 when the homologous chromosomes pair. And again, it contributes to genetic variation because genes that are usually next to each other might be separated, or at least on the same chromosome might be separated and moved to the other copy of that homolog. Remember, so here are homologous chromosomes. They are a pair because they are the same size and they carry the same gene information. When they align, they might trade a piece, a fair trade, and now our chromosome pieces are on the other of the homolog. Remember, they still belong, they're still in the spot where they belong, so it is not problematic. We might have a gene that codes for a particular trait higher on that chromosome that is no longer matched with its original. Therefore, we have genetic variation. This will contribute to variation within a family as well. Independent assortment is another way that we have genetic variation. Independent assortment occurs specifically during metaphase 1. If you remember, during metaphase 1, homologous chromosomes align at the midline of the cell, but it doesn't matter which order they are in. We don't have all the chromosomes that came from mom on one side and all that came from dad on the other side. They're randomly aligned as long as they are at the midline with their partner. So that leads to lots of possibilities when distributing these chromosomes to their resulting cells, into the gametes. So for example, even with just 4 chromosomes, we can see various combinations. So on the left, we have a possibility where we have 4 chromosomes. And in this case, the chromosomes that came from one parent are all on one side, and the chromosomes that came from the other parent are all on the other side. So when they are divided into the cells, we can see that all the chromosomes, the blue chromosomes, maybe they came from dad, end up in the same cell, and the red chromosomes that came from mom end up in the same cells. So we end up with 2 cells with the red chromosomes and 2 cells with the blue chromosomes. In possibility 2, we have 1 blue on one side paired with a red on that same side. Notice the homologous chromosomes are still lined along the midline, but we don't have all chromosomes on one side that came from the same parent. So now we have distribution that's different. Notice our resulting cells will have a variety from the original cell. Imagine if we're not talking about 4 chromosomes, but 46 chromosomes. The combinations will vary greatly and we will have very unique cells. So independent assortment, we independently assort chromosomes into the cells unrelated to where they came from. So an example of this is if we have, let's say we have a man that has brown hair and brown eyes, and his wife has blonde hair and blue eyes. When they have children, if the children inherit brown hair, does that mean they have to inherit the brown eyes? Of course not. They could inherit eye color from one parent and hair color from the other. Now, it's not quite that simple, but that's the idea. Finally, just a side note about when sperm cells do fertilize the egg cells. The fertilization is random. So whichever sperm fertilizes the egg is random. It's not determined beforehand. So whichever sperm cell reaches the egg and is able to fertilize that egg is the one that will carry the information for that offspring. Now, mitosis and meiosis are both cell divisions, but let's do a quick comparison. So in this slide, you can see the phases. Notice mitosis is 4 phases, where meiosis, we have double that. Our resulting cells also will differ. In mitosis, we have somatic cells going through this, so our body cells, skin cells, liver cells, etc. Meiosis is specifically for the formation of our gametes, so division of our sex cells. In mitosis, we have our chromosomes carry on individually, where in meiosis, our homologous chromosomes pair initially so we can separate them. Remember, crossing over can occur because those chromosomes are paired in prophase 1. In mitosis, the chromosome number is maintained. So if a cell divides by mitosis, whatever number of chromosomes we start with is what we will end with in each cell. Meiosis, on the other hand, we start with 4 chromosomes here in this case, but we're going to have a reduced number of chromosomes, so half, so 4 to 2. If this was human cell, we'd have 46 to 23. We go from a diploid cell to a haploid cell. In mitosis, of course, we have prophase through telophase only one time, so 1 division. And in meiosis, we have meiosis 1, so division 1, and then division 2, meiosis 2, so 2 divisions. The result of mitosis is 2 cells. These are called daughter cells. And the result of meiosis is 4 cells. Remember, these are 4 unique cells, not identical cells. So our cells at the end of mitosis match each other. They're identical genetically speaking, and they also are identical to the parent. Our daughter cells, or resulting gametes in meiosis, are unique. They carry varying combinations. They don't match each other, and they don't match the parent.